Hello friends, hope you all are well and doing good. Welcome back to my channel. In my previous video, I gave an introduction about nucleases and today I am going to take restriction endonucleases. So let's start our topic. Restriction enzymes, also known as molecular scissors or DNA cutting enzymes. They come under the category of endonucleases. They recognize a specific sequence in the DNA and makes a cut at or near that sequence. Now, the specific sequence that was recognized by restriction endonucleases in DNA is known as palandromic sequence. You can define palandromic sequence as a sequence that was read same on the two strands when the orientation of reading is kept same. Here you can see in this figure, one strand is this in 5' to 3' direction, the another one is in again 5' to 3' direction. When you read the sequence in 5' to 3' direction, it is GAA TTC, and on another strand again in 5' to 3' direction, the sequence was read as GAA TTC. So here you can see that on both the strand, when the orientation of reading was kept same, the sequence was read same. How restriction enzyme works or act? Let us try to understand this with the help of this diagram. Here you can see in this diagram, this is your vector DNA, this is your foreign DNA. Both these DNA has palindromic site. EcoR1 is a restriction enzyme which recognize this palindromic sites in both the DNA and make a cut not at the center but little away from the center at the palindromic site. This cutting result in the generation of an overhang stretch known as sticky end because they have ability to form hydrogen bond with their counterparts. In the next step, with the help of the DNA ligase enzyme, this sticky ends join together to form a recombinant DNA. Restriction enzymes are the indispensable tool for the recombinant DNA technology because they are used in laboratory to manipulate the fragments of DNA. Naming of restriction endonucleases. Restriction endonucleases was usually named on the basis of the organisms in which they were discovered. For example, HIND3 was isolated from Haemophilus influenzae. Now, let us have a look on the general rules for naming the restriction endonucleases. The general rules for naming the restriction endonucleases are as follows. The first three letters of the name are italicized because they abbreviate the genus and species name of the organism. Second, Roman numerals are used to identify specific enzymes from the bacteria that contain multiple enzymes. What is the difference between a sticky end and blunt end? As we all knew that in case of sticky end, a single stranded overhang stretch was generated or produced at the end of the DNA fragment, and these single stranded stretches have ability to form hydrogen bond with their cut counterpart. Whereas in case of blunt end, there is no unpaired bases or overhangs in the end of the DNA fragment, and this pieces of DNA cannot anneal to each other, hence more difficult to clone. With this, I am ending this video now. In my upcoming video, I am going to cover agarose gel electrophoresis technique. Please like my video, subscribe them and share them with your peer group. Till then, good day. Bye bye.